In classical Mendelian genetics, we learn that two genes assort independently and together produce the phenotypic ratio of 9 ratio 3 ratio 3 ratio 1 in a dihybrid cross. But in real life, inheritance is more often complex than what Mendel observed. Genes do not always act in isolation. Instead, they frequently interact with one another. One of the most important form of such interaction is known as epistasis. The word epistasis literally means standing upon. In genetics, it refers to a situation where the expression of one gene depends on or is masked by another gene located at a different locus. In this interaction, the gene that masks the effect is called the epistatic gene, and the gene whose effect is suppressed is called the hypostatic gene. The term epistasis was first introduced by William Batson, who is also remembered for coining the word genetics itself. It is important to note that epistasis is not the same as dominance. In dominance, the interaction occurs between alleles of the same gene. Like, for example, in pea plants, tallness is dominant over dwarfness. In epistasis, however, the interaction occurs between alleles of two different genes at a different loci. So, epistasis extends our understanding of hereditary beyond Mendel's simple laws. Depending on how one gene influences another, epistasis can be classified into different categories. Each type is associated with the characteristic modification of the Mendelian 9 ratio 3 ratio 3 ratio 1 dihybrid ratio. Let us look at the main types one by one. So, the classification includes four types of epistasis: recessive epistasis, duplicate recessive epistasis, dominant epistasis, and duplicate dominant epistasis. Now let's look at the types one by one. Recessive epistasis. It is also known as supplementary gene action with a ratio of 9 ratio 3 ratio 4. So in recessive epistasis, the homozygous recessive condition of one gene hides the effect of another gene. And a classic example for this epistasis is coat color in mice. So here, gene A controls the pigment production. The dominant allele capital A allows pigment formation, while the recessive small a prevents it, leading to no color, which is albino. And gene B controls pigment distribution. If capital B is present, the pigment is distributed as agouti, which is called grey coat. If small b is present, pigment is disposed uniformly, producing a black coat. If a mouse is small a with no pigment, it will be albino, no matter what gene B is. If capital A is present, then gene capital B decides whether it will be agouti with capital A and capital B or black with only capital A and small b. So when you do the dihybrid cross, instead of Mendel's 9 ratio 3 ratio 3 ratio 1, the ratio becomes 9 ratio 3 ratio 4. This is the Punnett square of this epistasis. You could easily see the ratio. This is because the recessive condition small a is epistatic over gene B. And remember, in exams, if you see a 9 ratio 3 ratio 4, it's almost always recessive epistasis. And you could use this trick to learn the Punnett square. You can make a triangle and a small square, and left is black. So the triangle is agouti and the square are albino and the rest are black. Now we will talk about duplicate recessive epistasis, which is also known as complementary gene action with a ratio of 9:7. So in this case, both genes are required to work together to produce a phenotype. If either gene is homozygous recessive, the same phenotypes appear. So this type is very interesting because it shows how two genes are both essential to produce a trait. If either one is missing, the final phenotype is absent. And a classic example for this is flower color in sweet pea, Lathyrus odoratus. So gene capital C produces a colorless precursor molecule, and gene capital P converts this precursor into final purple pigment. So the logic is, if a plant has at least one dominant capital C and one dominant capital P, the full pathway works and purple color will form with capital C and capital P. But if either gene is homozygous recessive, that is small c or small p, the pathway is blocked and the flower stays white. So the modified ratio becomes 9:7 with 9 purple and 7 white. This is called the complementary gene action because both genes are complementary parts of the same pathway. This is also shows how genetics connect to biochemistry. Genes encode for enzymes and enzymes work in a pathway. Here you can see the precursor with white flower, C gene enzyme, the intermediate compound will form, and if P gene enzyme is present, then anthocyanin pigment will form the purple color. If any of the enzyme, capital C or capital P are absent, then the flower will be white, and if both are present, then only the color will change to purple. Hence, this is called as complementary gene action. Next is dominant epistasis, which is also known as masking gene action with a ratio of 12:3:1. In dominant epistasis, the presence of single dominant allele of one gene completely masks or suppresses the expression of another gene. This is why it is often called as masking gene action. And a famous example is fruit color in summer squash, which is also known as cucurbita pepo. There are two genes involved, gene W and gene Y. So gene W controls whether pigment is disposed at all. If the dominant allele capital W is present, pigment deposition is blocked and the fruit remains white regardless of the other gene. And gene Y determines the actual color what pigment disposition is allowed. Capital Y produces yellow fruit while small y produces green fruit. So the outcomes are, with single capital W, it will be white. That is, W masks everything. With small w and capital Y, the fruit color will be yellow. That means no capital W, so pigment is deposited and capital Y decides the color. And with small w and small y, which is all recessive, the fruit color will be green. With no capital W and capital Y is recessive, that means small y is present, so the fruit will be green. So now let's see how this modifies Mendel's classical ratio. In a dihybrid cross, we normally expect a ratio 9:3:3:1, but because capital W is dominant and epistatic, all genotype contains at least one capital W. That is, 9 plus 3 is equals to 12 out of 16 results in white fruits. Only 3 plants are small w and capital Y, that is yellow, and one plant is small w, small y, which is green. So the modified ratio becomes 12:3:1, This is also called masking gene action because the dominant allele capital W masks the expression of capital Y. Now we'll talk about duplicate dominant epistasis with a ratio of 15:1. So in this case, either one of two dominant alleles is enough to produce the same phenotype. Only the double recessive condition gives a different phenotype. And a classic example for this epistasis is Awned development in rice. So gene capital A and gene capital B both independently control the production of awns. If a plant has at least one capital A or one capital B, it will produce awns. Only when the plant is all recessive, that is both genes are recessive, small a, small b, it will be awnless. So the ratio will become 15:1. This is something called duplicate gene action. Because the two genes provide a kind of a breakup for each other, and biologically this ensures that an important trait like awns, which protects seeds, is not easily lost due to a mutation in a single gene. Let's quickly summarize this. So the common types of epistasis and their modified Mendelian ratios are recessive epistasis with 9:3:4 seen in mice coat color, duplicate recessive epistasis with the ratio of 9:7 seen in sweet pea flower color, dominant epistasis with 12:3:1 seen in summer squash fruit color, and duplicate dominant epistasis with 15:1 seen in rice awns. So these ratios clearly show how gene interaction can significantly modify classical inheritance patterns. And epistasis is not just a deviation from Mendelian's ratio, it has important biological significance as well. So it demonstrates that genes function in pathways and networks rather than acting alone. Epistasis helps explain the biochemical basis of traits, since one gene may code for an enzyme that produces a precursor, while other codes for an enzyme that converts it into the final product. In plant and animal breeding, understanding epistasis is crucial for predicting trait expression. And in evolutionary biology, epistasis interaction influence natural selection, adaptation, and the genetic architect of the population. And epistasis is a fundamental concept that highlights the complexity of heredity beyond Mendel's simple laws. By studying epistasis, we learn how genes cooperate, suppress, or enhance one another, leading to a variety of phenotypic outcomes. Epistasis reminds us that the expression of traits is rarely controlled by single gene in isolation. Instead, it is the result of intricate interactions where one gene's presence or absence can completely change the outcome of another. Understanding these interactions not only deepens our knowledge of genetics but also provides valuable insights into evolution, breeding, and molecular biology. That's all for this video. Thank you.